you.
Thanks, Gabby. Again, I'm glad to be here to present the cellular engineering. I did last time also for Gabby, so I'm glad to see fresh and new faces. So today, my talk will be mainly on the biotechnology and cellular engineering. So how many people already have taken like molecular cell biology course before? Okay, many of them. So it makes my job much easier. So today, mainly I will cover 3 parts. Okay, basically one is isolation of cell and cell culture. Since if you already taken molecular cell biology, you probably already know a majority part of this section. And the second, not always. Okay, that's good. So in my talk also, if you have any question, just stop me. We want to make sure that you will follow my talks. So the second part will be mainly if you know how to do cell culture and study cells and purify cells, how do you analyze the proteins and molecules in these cells? What kind of technology are available for you to do so? Okay. And third will be mainly if you want to study this, what kind of molecular engineering or DNA technology you can empower to help you to study these protein functions or protein interactions. Okay? So I'll introduce some genetic engineering and DNA manipulation technology as well. So first, let's talk about how people can isolate cells. So we know in our body, it's whole mixture or whole mass, right? Everything mixed together. You can never know which protein or which cellular function are specifically performed by a single cell, right? Or specific type of cell. So the first thing for you, if you want to really understand the cells, you have to be able to isolate them and purify them, right? So that you know the function of a specific type of cells. So typically, if you want to isolate a specific cell from the whole mass or tissue, there's one way, basically, you can use so-called proteolytic enzyme to separate cell from extracellular matrix proteins. Because we all know in the body, all these cells are attached to a lot of matrix proteins and connect to each other as well, right? How do you select and separate these cells? This is the first step. So these enzymes can digest the cell membrane receptors so that they can be detached from the extracellular matrix proteins, okay? And when you add these enzymes, usually when you're doing cell culture, you probably know you usually also add so-called chelators for calcium. Why people add chelator for calcium? Mainly because the cell connect to each other, right? You have to detach cell from each other. And calcium is a major molecule help the cells to connect to neighboring cells. So if you chelate calcium, then cell will detach from each other. Okay? So this enzyme will detach the cell from the matrix. And the calcium chelator will detach the cell from each other. Okay? So with these two things together, the cell will become isolated single cells. Okay? So this is the separation. After you separate and detach, all the cells become single cells. You still need to collect a specific type of cell you're interested, right? And usually that's the several technology people use. One is centrifuge, so-called. Basically, based on different type of cell size, okay, or maybe their weight, you can use centrifuge with different speed, and you can collect different type of cells. Second is adhesion strength, because it's already known many different cells, they have different adherence strengths. So if you culture, for example, into the dishes for different time, the different type of cell attach to the surface. For example, for 5 minutes, probably fibroblasts will stick, okay? But other blood cells will take longer for them to adhere on the surface. So you can wash away, for example, after 5 minutes, you wash away, the other type cell will be gone. Only fibroblasts will be collected. Okay? That's based on adhesion strength. But all these, like the first one or two, the method are not that specific because a lot of different cell type, the adhesion strength could be very similar. So 5 minutes, you probably collect 2 or 3 type of cells. And the most accurate one is so-called antibody binding. If you already know certain specific cell type you are interested, and you know this cell type is expressing specific protein on the surface, you can use antibody to bind to it, right? Attach your antibody to a bead, and if you do centrifuge, and this specific cell type will be pulled down. Okay? So that's called antibody binding, and that's more specific. You can pretty much target a specific cell type you are interested. Okay? So these are general principles how to isolate cells, to purify cell. And these are the specific technology or machines you can utilize. One is called fluorescence activated cell sorter. Okay? How many people already heard about this fax machine to purify cells? Okay, very good. There are already people know. And the second I'm going to talk is about macro dissection. Okay? Basically, this is utilize laser power to cut the tissue and capture a specific group of cells. Okay? So this is the fundamental mechanism for a fax machine to work. Okay? So as we discussed, right, now you know how to add the different reagent to separate the cells so that they become single cells. Okay? So you collect the whole population of cell, and you can even add antibodies to label a specific cell type you are interested, right? And how do you select these specific labeled cell type? For example, you already have antibody with different fluorescence colors, right? To label the specific cell types, how do you separate them physically? That's the fax machine comes to play. So what you can do, you can have this reservoir system, and you can fuse in the cell suspension. Of course, this is a mixture of different type of cells, right? After you directly, purify from tissue. And there's a so-called sheath flow. Okay, it's wrapping up these, tubes. And these will start a flow passing through these long, narrow channels. Okay? During this pathway, there's laser shining continuously on this line, on this point. Whenever there's a cell pass here, based on the antibody labeling you already did, right? If it's labeled with specific type of antibody you are interested, it will label with different, charges. Okay? Whenever there's detection or antibody, it will label with a charge at this point. Okay? Because the flow and the distance are all precisely controlled. Okay? When you detect a cell you are interested, at this point it will tell based on the color or based on antibody it detected, it will assign specific charge. Okay? So now after you pass this point, the cell type you are interested is already labeled with specific charge. Okay? And they will pass this electric field. Okay? So you can imagine if they pass it, the specific charge of the cell will go to different tubes, right? For example, if the cell you are interested charged with negative charge, and it will go to this right tube. So you basically collect the specific cell type you are interested already. Okay? All the other things, if they are not charged, they will go to the central so-called waste tank. Okay? Is it clear for the mechanism how you can utilize fax machine to collect cells? Question?
Is there any limit on the size of the cells that can be separated using this process?
Yes, of course. So this will depend on the size, these nozzles. Usually like 10, 20 micrometer will be no problem. But if it's too big, like 100, maybe it will be difficult. It will stuck in the middle sometimes.
So what you're saying is that you're using the laser to ionize the antibody with which you label the cells?
No.
Correct?
No. So basically, laser is only serving as a detector. Okay. So laser, for example, shine, right, and you can see antibody labeling and color labeling, and it will tell this, detector analyzer. It tells, okay, there's a cell coming with specific, antibody labeling, and when they pass through and reach this point, it will be assigned a specific charge. So the charge and the laser are separated. This laser is detector, and this is, application site for the charges. Is it clear? Because this distance is fixed, right? And the flow rate is fixed.
So is the antibody fluorescent?
Yes, you can do that. Exactly.
And I'm trying to understand the meaning of the word fluorescence there. Where is the fluorescence in this whole process?
Yes, antibody can be attached to a fluorescence. That's the easiest way. So whenever laser detect a cell with fluorescence,
okay, so that's
right,
what you use.
antibody, and it will tell this site to apply a charge. Then this cell has to go to a specific tube. Okay?
Can you attach nanoparticles and use them with the fax machine?
Oh yeah, that's a perfect idea. Although it's not that popular yet, but definitely it can be done.
Because the fax machine, they think the nanoparticles will get stuck. Is there a size limit?
Oh yes, there's 10, 20 micron, so nanometer would be no problem at all.
So when laser like identify the certain cell, it
Yeah.
will label, for example, positive charge, and then it detects some other type of cell. It just put negative charge.
You can do so if you have multiple, color. For example, one specific cell type using green color, right? Another cell type uses the red color, for example, two antibodies, right? Two different antibodies. And when they go through these laser detect and find out green, it will give negative charge. If they see a red, they can give a positive charge.
How about like two type of cell just mixed together and?
You mean you want to collect two types of cells together?
Yeah, I mean you want to like separate
Yeah.
the cell, but
Right.
they are mixed together. So when,
Right.
when laser detect, they can detect like both, and then like how you determine how do.
Yeah, if, for example, there are two cells stick together, green and red together, right? It will detect, okay, it's a yellow color, for example, right? Mixed together. And they will not apply any charge and go to, directly go there.
Okay.
Only the single cell with purity will be collected.
Okay. Thanks.
Okay.
So the application of the sorting of the cells is it for cell count or is it for analysis afterwards? Because I'm concerned as far as like, is the charge sufficiently high where it can damage the cell, or is it still cell viable? Is the cell still viable afterwards?
Of course, cell will survive. Yeah. That's the way you can, identify and collect the specific type and continue to culture them. Yeah. There's another question?
So when you're talking about the detection on the other side of the laser, is it the steady state spectrum that you record, or is it a time resolved signal based on which you detect all kinds of?
Can you speak louder? Sorry, I didn't get.
So when we attach a specific fluorophore to one of the cells,
Yes.
what kind of signal are we detecting on the detection time? Is it like a steady state spectrum or like a time resolved signal?
Oh, okay. Of course, you can do, if you have a spectrometer, you can detect spectrum. But most of the time, it's just, a filter-based, camera. You can direct see the color, basically.
How about the width of the channel?
This channel?
Yeah.
This is about like 20, or 50, You can adjust, in fact, these nozzles. Adjust the size of these nozzles.
So you must know the size of the cells as a priori.
In general, you pretty much have pre-knowledge in terms of the cell size. Yes. But since you have this, adjustable size, right? And some kind of, knowledge of this cell population in terms of the size, you can adjust nicely, Pretty much, if it's micrometer range, no problem at all. Okay. Any more questions?
Why did you put the ultrasonic vibrator? What does the ultrasonic vibrator?
Oh, basically, just want to break, because sometimes cells are sticky. Even you add calcium, even you add these enzymes, right? They are separate from each other, but sometimes the membrane are still sticky.
Okay.
So, if you have some vibration to maintain their separation.
Okay.
Okay. All right, I'm glad. So I can move on, right? Okay. So another way is for some specific cell types, like Marina told you, right? For some cancer cells, they, grow together to formulate like a tumor type of thing, right? And in this cell, basically this is a tissue section. So for some specific tumor cells, if you are interested to study them, what kind of molecule signaling are going on in these cells, you want to study them, right? And there's one way you can easily do that without these complicated because majority of cells are not tumor cells. Only these specific confined region contain tumor cells. So there's one way called laser capture microdissection. Very simple. You can use a laser just to cut it, okay? And using this laser again to flip over to another dish, and then you can start to grow these purified tumor cells, and then study their functions, okay? So this is because, as you can imagine, if you use the traditional way, you mix up, like this is probably, 1% of the cell over the whole tissue, right? So that's a waste of the antibody or other technologies, right? Because you're only collecting this very well confined cell population. So laser capture dissection become more, economic and efficient, okay? So that's another way to purify cell populations. Okay, so after you go through all these, you can easily collect the purified cell types. That's what you get in the laboratory most of the time, right? You are culturing all these specific cell types. Although you probably didn't really, know how these cells are obtained. I'm telling you, that's, like if you buy cell line from ATCC, that's how they did it, okay? Through this fax machine sorting or laser dissection, So when you culture it, you can see these different purified different types. These fibroblasts, these are the myoblasts. They have very different phenotype. And this is oligodendrocytes and tobacco cells. Okay. Now you have purified cell types. You can really, harvest these cells, study the protein population in these different cell types. Okay. I think I just also listed the different cell types. I think most of you probably are working with some of these cell types, such as 3T3 cells. Basically, these are fibroblasts. MDCK also are epithelial cells from dog. HeLa cells, epithelial cells from human. Okay. Besides this purified cell type, there are another cell type I want to introduce is called hybridized cells. Okay. So it's a phenomenon that if you put two different cell types together, if you add specific chemicals, they can have some very interesting and weird phenomena happen. These two cells will be fused together, and their chromosome eventually will be integrated to become so-called hybridized cells. Okay? Most of the time this will not happen, right? This is a very rare case. You have to use chemical induce that. Otherwise, we will be in trouble, right? Our body have different cell type. If they all merge together, become one cell type, we are doomed, right? Mm-hmm. So this is a specific and very rare occasion, but this exists. Okay, if you introduce specific chemical environment, they can do that. So this cell type has very unique feature, and you can utilize to do some very interesting and cool stuff. One way is to basically produce antibody with very efficient or economical way. Okay, so if you want to produce antibody, that's typically what you have to do. You inject antigen into a mouse, and then the mouse will start to go through an immunoresponse and produce antibody, right? That's a typical antibody production process. But for example, these cells which produce antibody called B lymphocytes, they die quickly. Okay, basically you have to collect antibody from these B lymphocytes, but the yield will be very low because they die quickly. You can collect with a very short time window. Okay, and the number is limited. And people found out there are another cell type, which is tumor cell type or mutated cell type in this lymphocyte tumor. Okay, and these cells, they can grow forever, infinitely. Okay, and if you can put these two cells together and induce and create hybrid cell, okay, and some of the cell, they will become hybrid cell, and they can continuously produce the antibody you want. Is it wonderful? These cells will never die, and they can keep produce the antibody you want. Okay. And this cell is very interesting. You can, separate all these different cells and add some selection, medium, so that you can get a tube which contain only these hybrid cells. They will always grow, okay, and they can always produce antibodies. Okay, so this is the way people are producing antibodies with much more efficient way. Okay, utilizing hybrid cells. Please.
What's the proportion of the cells that becomes hybrid cells?
It's very low, probably 1% or below that.
But it's still much more efficient than just having mice.
Definitely, because after using this selection medium to select these cells, you can culture them and grow them forever. So you can like a big tank, right, to grow them, and they will continue to produce antibody. Okay. Instead, you have to go through this mouse, kill the mouse, and select like microliter or the antibody, every time, purify them. So that's really painful and low yield. Okay.
Is there any clinical proof that those antibodies created by this way will not induce cancer?
It's exactly identical antibody. The antibody you collect from the mouse versus the antibody you collect through the in vitro, they are identical.
Okay.
Right. Okay. You first
I
then.
have a confusion about the function of the antibody-antigen relationship. I misunderstood something somewhere along the line. I thought that the antigen provided the binding site for the antibody, like the epidermal growth factor receptor was the antigen, and the anti-EGFR was the antibody.
Right.
And so when you target the growth factor receptor, the anti-EGFR would bind to that site. I didn't know that antigens create antibodies too. Is there a dual function, or is there a misunderstanding on my part?
Okay, so this is of course not necessarily the hardcore this technology, right? But basically, whenever you have antigen go to the body, right, this B lymphocyte will be the one try to recognize this antigen, right? So they will start to synthesize antibody on the surface of this lymphocyte and recognize this antigen and grab it, right? And this is how you harvest this lymphocyte to collect antibody on the surface of this lymphocyte, right? But because in animals you only have limited number and limited time to collect them, the yield is low. That's why you want to create that. Okay, okay, sure.
Has there been any study where they've tried to maybe implant these hybrid cells for sustained drug delivery, like anti-VEGF production inside cells, inside the body, and are they viable inside the body, or do they need like some special media and stuff like that where they would only grow in vitro and not?
That's a very good question. I don't know actually. So maybe people are thinking about that's a very creative way, you know. Although I suspect though, these hybridized cells, because they are derived from tumor cells, right? They may be pretty, you know, cancerous. If we embed into the body, may not do, good things for the body. So I'm not sure, but I don't know whether people are doing that. But that's a very creative way to utilize this technology. Okay. All right. So we'll move on, and this is okay just to show you, that's modernized technology, to produce large-scale antibody, conveniently. All right. So now we know how to purify cell types, how to create these weird or interesting cell types. How do we study their functions, right? How do we study the proteins, molecules in these cells, in these purified cell types, okay? And this, I'm going to introduce a couple of technologies people utilize nowadays, right, to study them. One is the so-called centrifuge, because in the cell body, you can imagine, although the cell is purified, right, but there are probably millions of different kinds of molecules, proteins in these cells, right? How, again, do you separate them and collect a specific type of molecules, type of proteins to study their functions, okay? Centrifuge, chromatography, and polyacrylamide gel electrophoresis and mass spectrum will be the one we are going to introduce, okay? First, the centrifuge. Okay, this is a typical machine, so-called ultracentrifuge, can generate really, high speed. This actually, if you are working in the lab, you have to be very careful, because we heard some horrible stories. Because speed is so fast, if you are not correctly balanced this rotor, it can go off and generate a huge force, and this whole thing will just fly up. And when I was in UCSD as a graduate student, the floor upstairs, I heard some horrible story. The student didn't carefully balance the tubes, and the rotor just fly up and just damage the ceiling. Okay, you can imagine how much force it is. Okay, so the principle is, after you collect these cells, you destroy the cells using detergent, right, to break apart the cell and collect all the proteins, and use high speed to separate different population of cells based on their size, their density, okay, and separate the proteins based on their size and the density, okay. So this is basically the different speed to collect different kind of, intracellular organelles or proteins, okay. So for example, if you use a very low speed centrifuge, first you of course kill the cells, right, using detergent to break the membrane, collect all the proteins, and load into the tube and centrifuge. If you use a low speed, the bottom part, which is a pellet, mainly contain a lot of organelles, like a Golgi complex, right? Endoplasmic reticum, and some actin filaments, okay, cytoskeleton, nucleus, all will be pared down, okay. So this is the first layer. You can collect certain part of cell, okay. Now you take off the supernatant, which will contain some small organelles like lysosome, all these, and increase speed, okay. You collect another layer which contain like mitochondria, lysosome, or peroxisome, okay. And in this supernatant, you can collect this supernatant again and apply a further enhanced speed to centrifuge, okay. Now what you got is microsomes and small vesicles, okay. And you collect supernatant again, apply to so-called another tube, and apply ultracentrifuge speed, really high speed. Now you will collect these so-called like macromolecules, ribosomes, and this supernatant. What will be left in the supernatant in general after all these steps of centrifuge? Question? Cytoplasm and some molecules, right? Separated molecules in the cytoplasm. Okay? And then if you collect this supernatant, you can really study, the relatively small molecules or molecule complex, right? The majority, the big ones, has been separated, purified from different steps or centrifuge speeds. Is it clear so far? Okay.
Is this manual process, and also how long does it take to get to
this step, right? Probably, 1 day or maybe 2 days. Like this ultracentrifuge, you usually have to do like overnight. Yeah, because the size is so small, so going down is very, very slow. Okay. But anyway, going through this different speed of centrifuge, you pretty much separate the cell body complex, right, into different populations, right? Large complex versus small molecules. Okay. So these are the centrifuge. Another way you can also utilize is after you centrifuge and collect these different sections of the cell body, you can also use so-called sedimentation, either velocity or equilibrium sedimentation. For example, if you apply a collected pellet, okay, on top of this sucrose gradient solution, this is relatively with shallow sucrose gradient, you can apply centrifuge, and then you can separate these different molecules based on their size and shape, okay. Or you can create relatively sharper gradient of the sucrose and apply centrifuge, and you can separate these molecules, different molecules, based on their buoyant density. Okay? So in this tube, you can already see, different molecule will be located at different position, right? If you punch a hole on the bottom and collect different fractions, you will know, for example, this group of the different molecules will be collected at different tubes. Okay? So you separate different population of molecules by this way. Another way, you don't use sucrose gradient, you use so-called column. So what's inside this column? These are basically beads, okay? And you apply this mixture of the molecules on top and let them go through using gravity through these beads column, okay? And different molecules during this process, they will be separated based on their size or shape, okay? And because they are separated, if you collect, this solution flow through at a different time, you can collect different population of molecules. Is it clear? This is so-called, chromatography. Okay? And this chromatography can be designed using different ways. Basically, you can modify the beads so that you can separate different type of the molecules. Okay? You can either using so-called ion exchange chromatography. Basically, if it's ion exchange chromatography, that means the beads in this column are charged. Okay? For example, if they are with positive charge, you can imagine the molecules with negative charge will be relatively sticky to the beads, right? Right? Molecule with positive charge will be flowing through faster. Is it clear? Therefore, you can separate, right? The molecule with positive charge will be collected earlier, right? If they go through. And the molecule with negative charge will be collected at the later stage because they have become stickier to the beads. Okay? That's called ion exchange chromatography. And second type is called gel filtration chromatography. So for gel filtration chromatography, basically the beads are like sort of like broken, right? There's some, cavities on the surface of beads. So small size of molecule will be trapped. Okay? They will not flow through as fast as these big molecules because these big molecules cannot be trapped in these cavities. So they will be collected faster. Okay? Small molecule will be trapped and eluted later. Okay? So by this gel filtration chromatography, you can separate molecules based on their size, right? Large size molecule collected first, small size collected later. Question?
What is the typical size of these beads?
Good question. I don't know actually.
And obviously they have to be larger than the molecules
Yes,
themselves.
but in general should be like nanometer, 20, 50 nanometers. That's a little bit larger than the molecules, but close to the size of molecules. Okay? But these are not, as you can imagine, right? Many molecules, let me see. Many molecules, although here we only show different, like two kinds of molecules, but many molecules, they have similar size or they have similar charge. So these are not very precise separation, right? Molecule with similar size will be, trapped in these cavities, right? Most accurate one is antibody-based, and this is called affinity chromatography, okay? So if you want to specifically separate one type of molecule you are interested, you can code the beads with specific antibody, recognizing the protein you are interested. Okay? If you go through this column, then the protein you are interested will be trapped on the beads, right? Because the antibody recognize the protein you are interested. All the other protein will go through easily, right? So therefore, you can collect the protein you are interested with high precision using affinity chromatography. Okay? But in reality, in the cell, because so dirty, why we call dirty? Because there are so many different kind of proteins all messed up together, right? Only with one kind of chromatography is not enough to give you the purity you want. Usually people always combine them together. Okay? You go through multiple paths or different types of columns to get the purity of the proteins you want. So this is a typical way when you are in the lab, if you want to purify a specific protein, how many people already have done that kind of work? Purify protein from cells. Okay, so you must be good already. So these are very, basic principles for you if you want to start some new experiment, you want to purify some new proteins, that's how you do it, okay? First, as we already know, right, you purify the cell type, you break down, you centrifuge them, right? You already collect different type of the population of the molecules or complex. And then you load into the chromatography columns. Okay? Usually you will set up 3 type of columns side by side. Okay? The first usually you go is ion exchange chromatography. Okay? To first collect the population of molecule you are interested based on their charge. Okay? So here is the chart. This is the relative amount of protein go through. Okay? And this is the green line represent the concentration of the solution you add to the column. Okay? And when you add or increase the salt concentration, more and more protein will be eluted. Okay? And this pink profile represent the concentration of different type of the proteins. Okay? So when you add more and more salt, and eventually all the protein will be eluted. And this is the population of protein you are interested. And this is this section of the tube you will collect. Okay? So the first purification will be happening there. Your protein will be part of this population. Okay? After you collect this relatively purified protein, you load into a gel filtration chromatography. Okay? Run another column, and then your protein, your protein, right? The size relatively, right? And you can correct this population. similar kind of concept, but based on the size. Okay, this is based on charge. After you collect this is still not pure usually. Okay, usually they still contain multiple kinds of proteins all mixed together with similar kind of charge, similar kind of size. Okay, now you run into the last chromatography. Okay, affinity chromatography, which you coat the beads with antibody that you are interested, right? And then you elute all the protein which are not bound to the antibody. After you elute all these, then you add a really strong elution buffer, and the antibody and the protein you are interested in will be decoupled, right? The protein will be flowed through, and you collect this population. Okay. After these three type of columns, you will get relatively pure population of protein you are interested. Is it clear so far? Okay. If you go to the lab, you want to purify protein. That's usually the way you will, design your experiments. Okay. And after this purification, usually you will, if you run a gel, you can pretty much see there are, only like 2-3 types. Still not completely pure yet. Okay. But these are generally, depend on if you can collect enough of protein. Whenever you are using, chromatography, you usually need to collect enough of protein. If the protein amount is relatively small, then you are not qualified to do chromatography, because chromatography, you waste a lot of protein during the purification process. Okay? If the amount of protein is very, very tiny, then you need to consider other technologies, so-called, polyacrylamide gel electrophoresis. Okay? So the principle, how this SDS or beta mercaptan ethanol based electrophoresis is designed. Basically, these are the two, chemicals. They can bind to molecules and separate all the molecules to become relatively monomers. Okay. If the protein amount is not big, you use this so-called SDS page gel. Okay. You collect all the proteins because the protein amount is not enough, right? You cannot allow the luxury to go through the column chromatography. Then you directly run into a gel. Okay, collect all the protein, and these gel are connecting to reservoir. The upper reservoir and lower reservoir, these are serving as buffers, and these two reservoirs are connecting to different, electrode field. The top one will be connected to a cathode. The bottom will be connected to anode. Okay, top one will be negative charge, and bottom will be positive charge. Okay, and if you load the protein on these different wells, if you start the electric field, right, the protein will start to run through these gels, right? Which direction will they go? Down. They will go down, right? Why they will go down? Very good. I think I didn't spend enough time here. So you see these chemicals already mixed with the proteins, right? Molecules. So this chemical will wrap up the molecule, right? And the chemicals, particularly this SDS, it's basically negative charged, right? So you can imagine if this SDS is covering the molecule will be all negative charge covered. Is it clear? That's why this protein mixture together with SDS will be negative charge. If you start the electric field, they will start to run down, right? And you can imagine this is SDS treatment for the different molecules. You can imagine these two wells. On the left well, you can see there are two bands, right? This running, obviously, this is one population protein. This is a second population protein, right? These two population protein, they have different size, right? One run faster, one run slower, obviously, right? Through this running process. Which one has smaller size? Can you tell? The A has smaller size, right? Because they can squeeze through the holes in the gel easily because the size is smaller. Okay? So you can using this so-called SDS page gel to separate different protein populations, right? And then that's typical way people can use antibody to stain the gel to see the protein inside. Okay. This is the way how you use antibody to specifically stain the protein you are interested. Okay. Because as we mentioned, there are millions type of cells in the cell, or molecules in the cell. Even after you separate using SDS page gel, even you look at only one band, there are still multiple populations there, possibly. Okay? You have to use antibody to confirm the purity of these proteins. Okay? That's how people do it. So you already run the gel, separate different protein in the gel, right? And you assemble a sandwich, basically. You put a membrane, very tight, nitrocellular membrane, protein cannot penetrate. Okay? And you attach another two so-called filter papers. And usually you also add a sponge on top and on bottom. Then apply electric field. Top will be negative, bottom will be positive. If you start the electric field, what will you imagine? Protein will running from which direction? The protein in the gel, they will start to move, right? Where are they going to move?
So it depends on the direction of the magnetic field.
It's electric field. Top will be negative, bottom will be positive. So protein will be moving toward which direction? Bottom, exactly. So protein will get out of this gel, start to migrate toward the bottom and stick to this very tight membrane. Right? So you can imagine when you do this experiment, you have to be very, very careful. Some students, when they first start this kind of experiment, they made a mistake. They put the membrane on top. Then you can imagine all the protein will be gone, right? Because they migrate out and they would stick to the filter papers and get into solution. And all protein will be lost. So this is a very, very, very important step. You have to make sure the membrane is on the positive side of the gel so that the protein will get out and stick to the membrane. Okay? After protein is already collected, right, on the membrane, you can take out this membrane and add some antibody to it. But usually still you have multiple steps. One is you first so-called block membrane. You basically add BSA or non-fat dry milk to coat this membrane so that all these non-specific site on the membrane will be blocked. Antibody cannot recognize them. Okay? Then you add a primary antibody we called. This primary antibody will specifically recognize the protein you are interested. Okay? And then you add a secondary antibody which will recognize this primary antibody with some kind of enzymes attached. Okay? And then if you add some so-called substrate with chemoluminescence, okay, the enzyme on the antibody will react with the substrate, cause some chemoluminescence reaction. You will see the luminescence color coming out. Okay? So you can imagine this will be the typical Western blot. After antibodies, staining, you can specifically see the protein you are interested. They are in this position. Okay? The reason why you can see the color, because primary antibody bound to these proteins, right? Second antibody bound to the primary antibody, and second antibody has the enzyme to cause a reaction with the substrate to cause chemoluminescence reaction for you to see the color. Is it clear so far now? If you have enough of protein, you can use chromatography, right, to purify different protein populations. If you don't have enough protein, a little tiny amount of protein, you run SDS page gel to separate them and use antibody to recognize these proteins. Okay? Any questions so far?
Can you separate multiple components based on different colors in the chemoluminescence process?
For chemoluminescence, it will be difficult in general right now.
So it's only one color, right?
Yeah, for this traditional, biochemical assay, yes, it's usually just one protein you can see on the one membrane. But right now, there are some new technologies coming out. They can create different multicolor, Western blot. But that's relatively new. Okay. So obviously, you can imagine, in this kind of SDS page, okay, most of the time, these separation are based on their size, right? And use antibody to recognize this specific population. But in this spot, actually, they could still contain multiple population of the proteins. Okay? So people engineer another way using so-called, isoelectric focusing before you use SDS and separation based on size. So this concept is isoelectric focusing mainly is if you run this molecule into a solution and you create a different pH gradient in this solution or in this gel, okay, and if you apply electric field, like positive on top, negative on the bottom, this native protein with different charges. For example, this protein at low pH will have positive charge. This protein, the same protein at high pH will have negative charge, right? So obviously, if you apply this electric field, they will start to move, right? This guy will go up, this guy will go down. Until they go to correct pH, they reach equilibrium, they will no longer move, no matter what kind of charge, no matter what kind of electric field you apply. Is it clear? Because they reach equilibrium at certain specific pH. Is it clear? So these.
The media has different pH, so one is acid, one is base.
Yeah, sorry. This is not medium; it's gel. In the medium, you cannot create this, gradient easily because they diffuse easily. But in the gel, you can create the pH, different pH. You can maintain this pH.
And you put drops, for example, the protein on both sides.
Yeah. You can direct add, load onto this. On actually one side will be okay, even, because if you on this side, they will move down. If you add this side, they will move up.
So you don't need to have on both sides.
You don't need to. They will always go to this same position wherever you load, because they only can stay into an equilibrium side. Okay. So this is another way to separate proteins, right? Different proteins, they will go to different equilibrium, right? Because their native charge will be different. Is it clear? So you can combine these with SDS page. You first do this isoelectric focusing to separate the proteins, native protein, based on their charge. Then you add SDS and separate on another direction. Okay? Question?
So in this process, does the size of the particles play any role, or is it just the isoelectric potential?
In this case, no size will be important,
okay,
because these are native protein based on their charges.
And then you can use SDS.
Exactly. After you first do this, okay, based on this pH gradient to separate them in terms of their charges. Now you use SDS, add SDS, and run the SDS page, system to separate them based on size. So basically, you have two ways to separate them, right? That's so-called 2D gel. Okay. You can imagine. You first do this, right? On the on the X direction, you have very large gel. On the X direction, you create pH gradient, so the protein will be already separated based on their charges, right, along the X direction. After this is done, you start to apply SDS and run the SDS page electrophoresis on the Y direction. Is it clear? You first do this, okay? Charge will help you to separate different molecules based on their charge. But still, at each charge, there are multiple proteins together, right? Now, you separate on the Y direction, okay, based on their size. Now you can see this plot. Each dot will contain relatively purified protein populations. So far so good. This is purer, right? They're only using Wang. Okay. And then, for example, this is one, typical experiment. If you have tobacco cell, you just directly do this 2D gel running, okay? And the protein will be separated into different directions. And then you can use a Western blot, as we mentioned, right? Using antibody. For example, if in this case you are particularly interested in protein which are serine, threonine, phosphorylated, you can add antibody against phosphorylated serine and threonine, okay, on the protein. And you can see there are multiple dots here, right? The bottom gel is the protein which are phosphothreonine modified. Okay, so you can see among these millions of, I wouldn't say million, maybe thousands of different kind of proteins, there's only limited, protein which are serine phosphorated. Is it clear? So each dot, if you can match there, you can see this population of the protein are serine phosphorated. Okay? You really can, know in this specific cell what kind of protein. Actually, you don't know what kind of protein yet. You just know this size and this electro, property of the molecule is serine phosphorated. Right? You still don't know which protein exactly it is. Right? You don't know yet. Right? Now, how do you know it? How do you know it? If you already do that, and you know there are like 5 or 6 molecules are serine phosphorated, and you want to know which protein are they exactly. Do you know how to do that? How to find them?
Use antibody.
Yes, you already use antibody, and you already find out, okay, these proteins are serine phosphorated. But you don't know which antibody you should use, right? Because you don't know what are they. How do you find out? Exactly. That's exactly the way you should do. So the principle for mass spec is very simple. Basically, you have a protein, right? You can, for example, in this gel, you can basically, if you already run all this, and you can just cut out the protein from this spot, right? And you can chop this protein using enzymes. These enzymes, a lot of times, they will recognize lysine in the protein, so they can be chopped into like multiple pieces. Okay? And if you run like 2D gel or whatever, you can create a profile, okay, of this specific protein. Okay? And the lucky thing is we are living in a modern society, right? So there's millions of, the work has been done. People already chop all these proteins and already create a library. So they know exactly which protein has different, which protein has specific profile of these kind of features. Is it clear? Now you have MALDI, right? Mass spectrum, okay? You have run this 3D gel, and this dot you already identify. For example, this is phosphorylserine modified, right? And you want to know the protein, you directly cut this protein out. In the gel, you can cut it out, right? And chop it using different enzymes, and it become multiple pieces, right? And you run mass spectrum to find out abundance of the different pieces. Okay, you will get a profile. Is it clear? After you got this profile, you input this profile into a library and screen to see whether this profile match existing profile protein. Just like a fingerprint, right? If somebody got caught, they will get the fingerprint, and the FBI will directly key in this fingerprint and run through the library, right, to see whether it match to an existing fingerprint, and they will know this fingerprint belongs to this specific person in the library. Is it clear? Same thing. You run, cut this protein out, chop it up, become multiple, small pieces. You create a profile, right? And this profile will be keying into the library and start to screen and to see whether it match to an existing protein. Okay? Because existing protein already has been chopped and create profile, right? If these two profiles match, this protein, you can know the identity of this protein in the library. Okay? The assumption is all the protein in the cell has been already, purified and chopped and create this profile there. Right? Is it clear to you? Okay.
Just curious, how large is this database? Like, how many different kinds of proteins do you have?
Right now, it's pretty much every single protein, in the cells has been already created in terms of their profile. Right?
When you say that you're using enzymes to chop this protein,
yes.
You're talking about
protease.
Separating. So is this one single type of molecule now that you're breaking into subcomponents?
Oh,
or are they different molecules that you separate using enzymes, and then you run them through the mass spectrometer.
Very good point. So I'm assuming after you go through this 2D, right, gel, if you just directly cut out this spot, it's only one single type of protein. And you just chop it into different fragments like peptides, right? And run the profile and you match it. But of course, in reality, we all know, right? It's just ideal case, right? Sometimes, even if you cut one spot, there are multiple proteins there, okay? But still, you can cut it using these proteases to cut into different peptides and run this profile. And you can, this, I don't know how many you have already pre-experienced, in terms of how to match profiles. For example, this profile unknown protein, right, is the combination or multiple profile of the protein in the library. You can easily find out as well. Of course, the ideal case is, okay, this spot only has one single type of protein. Then your job will be very easy, right? You create this profile, run the library, match. Okay, you know the identity of this protein right away, right? But other times, this spot has like 2 or 3 type of proteins, right? You chop it, you create a profile, run the library, you find out none of them match. But if you do a little bit more investigation, you can easily find out, actually, if you combine 2 protein profile together in the library, it match your profile. Then you know this spot contains 2 known protein in the library. Is it clear? Of course, this probably require, a real analysis between these profiles, right? Question?
Does it ever happen where a combination of 2 proteins in a single spot generates a profile that matches another type of protein?
That's unlikely because all these. Let's assume all the library proteins, right? They are single proteins. People already purified and create this library. Okay. Any questions so far? So I guess after this, you are really a master, right? You can go to the lab, jump into it, purify proteins, purify cells, right? And you can identify whatever protein you are interested, right? If you combine all this together, right? If protein is large, you can use chromatography to collect the pure protein population. If protein you can obtain is only small, you can use SDS page and 2D gel separation to identify these proteins, right?
I imagine that your purity with this method will also be pretty bad because transferring that onto a membrane is probably going to be very difficult.
Okay.
Able to extract that spot.
Yeah, that's a very good point. Very, very good point. So in general, if you're really doing that kind of study, right, you don't do it just at once. Okay. What you do is you don't transfer everything to the membrane. Still, the majority will be left in the gel. That's is a tricky thing, right? But in the reality, you can do it in the lab. You can run this Western to transfer a small portion of the protein to the membrane, and you stand, right, and identify this spot using antibody, and you match this membrane to the gel back again and cut out the spot in the gel. Okay? So actually, the purification or the protein purification is still from the gel, not on the membrane. Okay? Membrane only will serve as a guide for you to find out or identify the spot where the protein is localized in the gel.
But if this is an ideal spot, it's pretty spaced out from anything else or any of the other spots in the top right.
That's a very good point. In reality, you have to, probably you have to collect anything spot, right? You can run another gel again to separate, right? Most of the time, when we are getting to the lab, that's why people can publish paper like 2 or 3 years, one paper, right? Not like 2 or 3 weeks a paper. Mainly because the reality of these protein purities, right? So, for example, if you are particularly interested in protein in this corner, yes, you have to cut it out, run another gel again to separate further. And of course, it will be purer, right? Because you discard all the rest of the protein will not create the background as bad as this gel. Is it clear? Many times you have to run multiple times to get the real purity of the proteins. Okay? So this, you pretty much know the cell and the proteins, and because the revolution of molecular cell biology, and now they can also provide very powerful tools for you to manipulate cells. Okay? Not only you can directly analyze and discover what's going on in the cell and in the proteins, you can also have the power to manipulate cells, manipulate molecules. And this manipulation can provide you a lot of power to identify certain specific functions of the molecules. Okay? Which I'm going to introduce some basic principles. I think most of them you probably are already clear, such as like PCR, polymerase chain reaction. How many of you already know this? Many of you, right? Okay. So this is basically, I think Nobel probably also added the, the fame for this technology, right? 1993 Nobel Prize. And this is actually the main inventor, Morris. He's a very interesting guy, He's in San Diego, basically really a playboy, type of person. Surfing is a major job of his, life. And actually, when the Nobel Prize was announced, he was also, on the board, surfing. So anyway, that's the technology. Basically, the fundamental thing is DNA. Okay. Before the PCR is generated, people always have difficulty because we all know whenever or whatever you are doing the technology, amount is always the key, right? Amount is always the key. If you don't have enough amount of protein or enough of DNA, it's always difficult for you to do anything in general. Okay, and this guy is really genius. He invent this technology to amplify DNA segments whichever you want. Okay, the idea is you have a DNA double strand, you know that, right? And if you want to amplify a specific section in the middle, you can design so-called primer, a small short piece of DNA. They can be matching to these two sides, okay? And use a high temperature to separate this DNA template and allow lower temperature again to allow the DNA primer to attach to them and add, DNA polymers and ATP, GDP, CTP, whatever, and synthesize the protein, new DNAs, okay? This is so-called first cycle. You can repeat this cycle, okay? First cycle, and that second cycle is after you synthesize from the first cycle, you separate them again and do a second cycle. And you can see already this DNA template, this black thing, they remain the same, right? Number, right? Always 2. And you can see already these, and these, and these, they become linear increased. Second cycle, you have 4 copies. And the segment which you are specifically interested, this segment and that segment, which is encompassed by the section you designed, only have 2 copies at the second cycle. Okay? Magic happen in the third cycle. You do the same thing again, okay? Amplify again. You will see after third cycle, okay, the DNA template, the parental DNA template still remain 2, right? And this segment, which has some kind of one side overhang, 1, 2, 3, 4, 5, 6, linear increase, right? Third cycle, 2 times 3, 6, right? And look at this segment which you are interested in, yellow highlighted, this 1, 2, 3, 4, 5, 6, 7, 8. It's already 8. Okay? This same magic will happen if you run another cycle. This segment will exponentially increase. Okay? And this segment with like little one side overhang will increase linearly. And the parental DNA will stay the same. So if you can imagine, if you run like 30 cycles, what's the majority of the DNA you will generate?
That's going to increase DNA.
Exactly. So this type of DNA will be exponential increase for 30 times, right? So after you finish the 30 cycle of the PCR, the most majority of the DNA you collect these. Other things are really negligible, right? Because this will be linear increase, not much, right? This will remain the same. Okay? So PCR can allow you to amplify any DNA segment, okay, in a short time with large quantity. Is it clear? Okay, so this is PCR, which will allow us to collect enough for DNA. Another technology is so-called restriction nucleus. Okay, so this is also Nobel Prize 1978. Also provide huge power for the molecular biology revolution. Basically, the principle is they found out these specific enzymes which can cut the DNA. But the good thing is each enzyme will cut and leave a very unique interface between cutting site. It's just like in Legoland, right? Or a puzzle, right? It creates specific, interface. That's how you can piece the puzzle together again, right? Because they have unique interface. Each enzyme have their own unique interface. Okay? You can imagine after you cut, only this specific site can be, pieced together. So if you use the same enzyme to cut 2 DNA fragments, piece them together, you can connect 2 different DNA together, right? Because they have the same interface. Okay? And there are different enzymes, DNA enzymes which can help to join the DNA together. This is not as important as this, nuclear restriction nucleus, which can create specific cutting site. But this also provides power for you to, after you cut, you can join them. Okay, if they are cut by the same DNA restriction nucleus. Okay. Now with all these together, you can create enough of DNA. Then you can insert them into, so-called circular double-strand DNA, and this DNA can be put into bacteria. We are going to show, you can do a lot of wonderful things. For example, you have circular DNA. You can insert a DNA fragment using PCR to amplify, cut with restriction nucleus, right? And if you cut using same restriction nucleus, they will have same site, so that you can perfectly match, and using DNA ligase to put them together. Is it clear? Have to use the same restriction nucleus. If you use different one, you cannot insert. Okay? Now, with this kind of engineered plasmid or DNA, you can put into bacteria. The bacteria continue to grow and amplify the DNA. Now you got tons of DNAs, right? But that's not about it. There are some other things you can also, enjoy. Basically, after you engineer these DNAs, okay, you can put into bacteria. They will not only amplify DNA, they can also allow you to produce the protein you want. Okay? So using genetic engineering or DNA technology, you can pretty much engineer and create large quantity of protein, whatever you want, because all the protein are controlled by gene sequence, right? If you know the gene sequencing, using PCR to create the DNA, insert into this plasmid, you can create the protein you want with high purity and large quantity. Is it clear?
How do you put the DNA inside the bacteria? Is it like injection or diffusion? How?
That's a very good point. So this is a transformation. I didn't describe this here. But in general, you have bacteria, right? You mix them together with the DNA you already create, right? And usually people do a so-called heat shock. So the membrane of the bacteria become permeable, and the DNA will diffuse inside the bacteria, okay, and start to, incorporate into the bacteria system, okay, and bacteria can grow and amplify DNA and the protein you want, okay. Now you know the DNA technology. You can use DNA technology to manipulate the genes, right, to create the protein you want, okay. What kind of wonderful things you can do with this technology? I want to introduce a couple, and then I should be done. One first thing is, for example, if there's protein, okay, but this protein you know the gene sequence, but you don't have antibody available for it, because antibody, you have to rely on a company, right, to create the antibodies, right? If there's no such antibody available, what are you going to do? Of course, you can say, I will just generate antibody. That, I can assuredly tell you that's painful, and expensive. Your, advisor probably will not be able to provide that kind of resource for you to create specific antibodies, okay? Then genetic engineering can help you, okay, to amplify or purify this kind of protein in your lab, okay? The way is you can easily engineer the gene sequence, because that's, you can easily, download from the PubMed, and then insert a known, gene sequence which can create a very cheap peptide which can be recognized by commercially available antibodies, for example, his tag or flag tag, Then you put these two together, and then put into bacteria and produce the protein. So this protein has the protein you are interested, also has the epitope tagged peptide, Now you don't need to generate antibody. You can direct all the antibody from commercially available, resource, and this antibody will recognize this tag, right? And will pull down this whole protein out, right? You can put the beads onto the antibody. The antibody will bind to this hybridized protein and pull out this protein. You can analyze the function of this protein. Is it clear? You don't need to create antibody in your lab. That's one way. Already help you a lot, right? Another way is if you want to study the protein-protein interactions. For example, you want to know whether protein A or protein B are interacting to each other. In this case, protein X and protein Y are interacting to each other in the cell. How do you do it? You cannot directly use antibody, right? Because the current microscope resolution is 200 nanometer, right? And the protein obviously are much smaller than that. If you use two-color antibody to label these two different proteins, if they are in the same place doesn't mean they are interacting to each other, right? It's just like two protein or two person in the building doesn't mean they are in the same room. So I have limited time. I will just quickly go through. What you can do using genetic engineering is you can using DNA, like encoding a blue frozen protein together, so create a hybrid protein, and this green frozen protein hybrid together, and these two proteins you can put in the cell. If these two protein XY doesn't interact each other, you excite this blue frozen protein, you will see a blue color. Only after these XY interacting to each other, exciting the blue frozen protein, you will see a green color out. If you see a green color out, you know these two proteins are interacting to each other. If you see a blue color out, you know these proteins are not interacting to each other. Very simple, right? Genetic engineering can help you to do that. If you are interested in one protein, again, protein X, you don't have antibody available for it, and you want to study the protein interacting with this protein X, how do you do it? Genetic engineering can help you to do that. You fuse a GST tag to a protein X, and there are antibody available directed against GST, and these beads and the antibody can bind to this and pull down this protein, right? And there are a lot of protein, of course, associated with the protein X already. And when you pull down this whole thing, the protein associated with the protein X also being pulled down. You can analyze this protein associated with the protein X, although you don't have antibody against protein X. Or if you want to study a protein interaction, you can also using so-called yeast-2 hybrid system. Again, these bait and play are the protein which controlling the gene regulation. And this protein, let's say X and Y, you want to know whether they're interacting to each other. You can create this using genetic engineering, create these two hybrid protein. This bait will bind to DNA. This prey will start to, regulate the gene expression. Only when these two proteins interact with each other, they will be functional to create protein. Colored protein, for example, you can generate. If they don't interact with each other, they will not create this reporter protein. Another case, right? You can use genetic engineering to help you to study whether these two proteins interact with each other. I think last case, I think the most wonderful, application of genetic engineering is so-called phage display. For example, if you have a specific tumor or cancer you want to detect or you want to inhibit, how do you do it? Now people can help you do that. You first using genetic engineering to create a library of the genes. People can do that easily, millions of the different genes encoding different peptides. And they can be expressed into, for example, E. coli or yeast. They will be on the surface, so E. coli or yeast. So you can imagine if you create a library of the DNA, you can create a library of yeast or bacteria Each single bacteria or single yeast contain a different peptide on the surface. Is it clear? Millions of peptide you create. Now you inject into mice, okay? Only these bacteria or yeast express the intacting peptide will stay in the tumor. All the other yeast or bacteria will be washed away in the body, right? If they don't interact with the tumor. Is it clear? Now you harvest the tumor, you can extract the bacterial yeast. You know these bacterial yeast, they interact with the tumor, which means the peptide on the surface of this bacterial yeast are interacting with the tumor. Okay? And if this tumor becomes shrink, you know this peptide can specifically inhibit the tumor shrinking. Okay? And you know what kind of gene encoding these peptides, right? And you know what kind of gene sequence or peptide sequence will be a good drug to kill this specific type of tumor. You can certainly isolate these bacteria or yeast, correct the genes, and do another round of mutation, create another library, millions of library. Okay? So these already have some certain effect on the tumor, right? They can cause shrink. After you create another library, based on these genes, okay, you can create another library and inject again. And one or two of them will be more efficient, obviously, right? Some of them not as efficient as the original parent gene sequence, but some of them will be more efficient. You can do another screen until you find a best peptide or best gene sequence encoding peptide which can directly kill the tumor. Right? I'm not sure how many of you are interested in cancer therapy, but that's a very typical modern way for you to do so. You can create the tumor in the mice, create a library, inject, okay, select the one which bind to them, inhibiting them, and do another gene mutation to create another library. Going through multiple cycles, you will find a very good efficient peptide and gene sequence which can kill the mice tumor. Okay? These are all based on genetic engineering. You have to be able to generate different kind of gene sequence and generate different library of these gene peptides. Okay? I think that's my last example. I think because of time, I will not go through details anymore. If you have any questions, I will be happy to discuss with you afterwards. Okay?